Welcome, everybody. Today is the last workshop on the Avia Online Workshop series for this current run. Today we're very happy to give you a quick kind of rundown and demo of the newest recipe that we just released back in June, the 3D cell analysis. And since most of you have already been to our previous workshops, I'll just go through this pretty quickly. So as you know, Avia was launched back in 2017 in April, and over the past 3 years, we've been continuously adding new features to the product. And so and ending up to Avia 9, which was just launched in the middle of last month, which we have with the cell analysis, with the new transparency options and everything. So today I'm really happy to show you guys what the 3D cell analysis. And then of course, as you know, in terms of the Avia product family, we have a couple of different products. So we have Avia, which is our main product, which runs on the desktop. Avia Cloud, which allows you to train deep learning models in the cloud. Avia Web, which is what we're going to be using to do the demo, to do the workshop today. So you can get to play around and have full access to Avia, all the features of Avia, right from your browser. And then, of course, we also have a free product called Avia Community, which allows you to view 3D data, but also to create video and also do pixel classification as well. So in terms of how we got to the 3D cell analysis, we have to think about the different scales that exist in biology, starting from the very small, very fine, very granular level. So if you have a very good microscope, you can get to see the cell, the very specific cellular machineries, say, for example, the nuclear pore on the very left. And of course, then of course, those nuclear pores are part of the nuclear membrane that is composed of the different organelles, like the nucleus or the mitochondria and everything. And then of course, all of those organelles then get organized or are into a higher level called what we all now know as a cell, which has the nucleus, the mitochondria, and all these different types of vesicles. And then further up, once we look on in this, we have to going from the cells, they organize into different tissues and different organ systems, and then that's also another layer that people are very interested in looking at. And of course, then at the very end, we have the tissue that and all the tissue combined to make an organism, which can be as simple as something like a yeast cell, but all the way very complex up to mammals and humans and all and other animals essentially. And for our and in Avia, what we've done is to create a system with the 3D cell analysis that allows you to analyze not just the individual components as a standalone, but also creates the relationship between the different objects. So we have, starting from the very basic level, we have these cells and organoids that I'm going to show you in the demo later today. And then of course this is up to a full embryo, which then have a lot of small cells in the middle of it. And of course, then all of this can also be further expanded to the whole organism. Sorry, that allows us that we can analyze with this this recipe. So even though it's called the cell analysis recipe, it doesn't the recipe itself is very flexible to the point where you can actually as long as you have anything that can be considered a big compartment with little small things in it, you can actually use the recipe to create to analyze all of those individual components and then relate them so you can get measurements like how close or how far an object is to the edge of the cell or edge of the embryo, for example. And of course, I'll have a little bit more about the measurements in a little bit as well. So in terms of the 3D cell analysis recipe. This is the interface. It looks very daunting in the beginning because we have everything turned on. Essentially, we have a group for the whole cell detection. Then we have nuclei, and then we can detect up to 5 different smaller compartments. And of course, like there could be more compartments that may need to be analyzed, but for now, we're just going to keep it like kind of a little bit more simple and just keep it up to like about 5 or 6. And then, of course, like I said, this is the full interface, and there are options in the input and output where you can adjust which object you want to, like what you want to detect and how many objects you want to detect. So you can turn things off and on, and it's pretty flexible. And in the upper right corner, this is something that is new in Avia 9, and also this is very new for the cell analysis recipe, is the ability to pick the threshold parameter automatically. So you can either set press the button to set the parameters either on the automatically for the entire image, or you can also create a small ROI that and have the software kind of quickly figure out what the best parameter for segmentation is. And of course it's very adaptable to different input processing that you apply to it. So if you add a smoothing, or if you add remove background, it will automatically adjust the best threshold parameter based on those options that you've selected. And then here, this is one of the object set detections. So there's 3 different options that you can choose from for each detection group. The first one is the smoothing, which we have 4 different types of smoothing plus the skip smoothing that allows that sometimes like when your image is kind of grainy, it's good to kind of like apply a little bit of smoothing so that you can get a better detection result out of it. The next one is the background removal. So you can choose to remove the image background or skip it so that you can have a cleaner image that you can use for detecting the objects. And then finally, at the very end, we have the partitioning options, which allows you to essentially separate the objects that are touching each other. Or if you have, like, say, for example, if you're detecting a whole embryo or a whole organ in the whole cell detection, you can just skip the partition because then you can analyze everything within just one massive tissue or object that you're looking at. And then for the nuclei, there's also an addition nucleus, or you can also extend the cell boundary to include everything that the nucleus includes, or crop the nucleus. Because sometimes, in terms of in the imaging, you might not get the quality of the result to actually accurately segment both the cell and the nuclei within the same region, and so that's why we have these options so that you can correct the appearance so that your cell looks more realistic, so to speak. Then see. And here's just a quick video of how just everything works. So you have the input. You can create, open up to a preview of our quick ROI, and can see what the result looks like. And then if it looks good, then you can obviously go on with this. And then another thing that this is showing you is the transparency option that you can actually see, so that you can look at objects that are both inside and inside the cell while you're looking at this. And yeah, so this, let me just skip right past this one because this one in this video, this is showing just a lot of the visualization options that we have in Avia. Okay, so. And so what's really nice about this 3D cell analysis is that we are also introducing a lot of new measurements because we're looking to look at to apply a multi-scale analysis to the image. We need to have ways to look at how each object relates to each other. So we have, of course, we have like standard things like the volume ratio between different compartments. Of course, the intensity is another one. Position is the one that I just started to mention. And it's really nice, especially for things like looking at exocytosis, where you want to look at how close the vesicles are to the edge of the cell and see whether they're likely to be undergoing exocytosis or if they're or something else is going on. And then, of course, you can get a count of the organelles in each cellular compartment. So you can count the number of vesicles within the whole cell. And you can also count the number of vesicles in the nuclei, and you can generate a ratio between how many of those vesicles are in the nuclei versus how many of those are in the cytoplasm area to get a sense, to get additional information about the objects that you're looking at. And here's another video. So today I'm going to show you two different examples. So this first one is a Drosophila egg chamber. And so for this one, we actually detect the whole cell as the egg chamber itself, and then the individual nuclei inside the egg chamber as as a vesicle. And so for this one, like if you look earlier, so you can see that there's some nuclei that are they're smaller, they're on the closer to the edge of the cell, to the edge of the egg chamber membrane. And then you have the bigger nuclei that are sitting right in the middle of the in the center of this egg chamber. And then another one that I'm going to show you, this is a gastric gland organoid image that's taken from our good friend at Free Eye at La Chica. And in this one, there's lots of things that you can do with, in terms of the analysis. So in this one, we counted 16 cells and some around like 1,600 vesicles. And there's an average about 98.8 vesicles for each cell. But when we look closer into the data, each individual cells actually have different number of vesicles. So you have some cells like cell 7 and 6 that have over 150, 146 vesicles within the cell. And you have some that have very few. So like cell 15, which only have 20. So these kind of variations could be important to look at at an individual cell level. And when we aggregate the data all together, it's something that that can easily be missed. But with the 3D cell analysis, this is something that you can look at more closely and actually be able to extract that information with. And here is one of an Drosophila whole embryo. And I'm not going to show you this one for the in the workshop. But what's really cool about this one is that this is an embryo. This is a whole fly embryo that is taken by Philip Keller, and we have generated a couple of different interesting regions of detection. So this green stripe down the middle is the center line that we have detected with the cell analysis, and then you can see how the objects are kind of how far the individual cells are from this center line region. And then also we have also done a detection on the tip of the embryo as well. So there's lots of things that you can do in terms of your analysis with this recipe. And there's just a couple more images of that data set that we were just looking at. So of course, like on the left, you have the volume rendering that we have done that has been in Avia since Avia 5, really. And then of course, object segmentation, which we have been able to do previously with just the 3D object analysis recipe. And everything right now, I think, is color coded by the measurements. And then the two new things that we added to this particular analysis is the tip of the object, and then also the center line, and also the center line of the object. And you can see the objects that are visible in each of these individual regions. And then we can all put it all together and kind of show how each of the cells are related to the tip or to the to the center line. And here's like some of the measurements that we generated using the analysis, the recipe. So you can see there is some variability in the objects themselves within the different regions of the Drosophila embryo. And so, of course, like I think that I've kind of like went through this like pretty quickly. So hopefully, if you guys have any questions, like do feel free to ask anytime during the presentation after this during the demo. So I do want to thank the Avia team for making all of this possible. Of course, like excluding myself there's like the top 2 rows are software and algorithm engineers. And then actually the top 3 rows, most of them are software and algorithm engineers. And of course, we're also looking for 2 new sales and application specialists, one in the Europe region, and then also one in the northeastern US. So if you know anybody who's looking to transition looking for a job, do kind of just pass the word along. And of course, none of this work could be done without our research collaborators. So here are all of them. I'm not going to go through all of their names, but each one of them have been like very instrumental in helping us kind of build up our system and like build up all of the analysis capability in Avia, and providing us the valuable feedback in terms of how to make Avia more accessible to people, to researchers. So, and so like that, it's the, and so like the next thing we're going to talk about is like, let's get right into the workshop. So a little bit of info before we start. Everybody is going to get a unique AppStream link, and you should be getting that in the chat window for that Quinn is going to be sending out. One thing to note is that please only click on the link that you're assigned to. If you click on somebody else's link, you can actually see what they're doing, and then you guys have to fight against each other for control of Avia Web. And so we want all of you guys to actually be able to do your to play around with Avia. So click on your own link, and then there's a toolbar in the very top. Sometimes like it can be a little bit laggy between what your mouse is seeing and what Avia is doing, because you're connected to a web server that is somewhere in that is somewhere away from basically it from where your institution is. And then if you have issues with the with Avia, like part of Avia getting cut off, you can click on the little gear icon and change the screen resolution to match your display, or make it smaller or bigger. And then finally, once you get into Avia, this is the interface. So for the most part, what we're going to be focusing on is going to be in the second section, the data viewing and exploration. And then, of course, the recipe console area, which we will, which on the right, you can actually change the layout a little bit and to make it more accessible to you. So as I mentioned, we're going to go through 2 different exercises today. The first one is the gastric gland organoid image. It will try to detect the cell, the nuclei, and 3 different types of vesicles. And then finally, and then we'll end the presentation with the Drosophila egg chamber, where we will detect the whole egg chamber as our whole cell in the sense, and the individual cells as vesicles in this case. So thank you very much. So right now you should all have received a link to the app stream. So let me switch over to the. Okay. So when you open that, when you click on your link, this will actually launch this window, and you can just to start. We can click on Avia 9, and then it will take about 2-3 minutes for Avia to run. If you have data that you wanted to upload or transfer ahead of time, you can always you can use CyberDuck. So, but today we're for the workshop, we're going to try to focus on the Avia 9. So it takes usually about a little under 2 minutes for Avia to launch. And so we'll at this point I will just stop talking and then just while this launch. And if you guys have any questions, feel free to unmute and ask it. Or if you want, you can also send mention it in the chat window as well.
Is Avia Web asking for a serial key from everybody? Because it looks like both Heather and Alex, you're experiencing the same issue.
Yeah, it asked me to click on activate, and I did that twice, but now it says unable to validate serial key, unable to establish connection.
Okay, Alex, I'm gonna send you a different link right now. Give that one a try and let me know. Heather, is it doing the same thing to the second link or the first link?
It was the first link.
Okay. Alex, did the second link work for you?
It's still loading, sorry.
Okay, no problem.
You can go ahead. You can go ahead. I should be able to pick up.
Okay. Yeah, so this is the Avia web window. And today, so the demo data is already uploaded to the server already. So if you go to File Open, we will go to the demo data. If go to the Share Data folder, and then go to Demo Data and 3D MSA. And we will start on the 3D cell analysis demo image. So Alex, is Avia Web loaded for you already?
It's activating. No, it doesn't want to activate. It's the second link.
Okay.
All right, Alex, I'm going to give you another link now. This one is from the US West. So things might be a little bit slow for you, but let's see if that works.
Okay, thank you. You should go ahead.
Yeah, okay. Yeah, so this is the data, this is the gastric gland organoid data set that that we kind of been showing quite a bit on with our presentation and in the demo. So for this one, what we're going to do is we're going to detect the cell, the whole cell object, and then also the nuclei, which is the nuclei, the vesicles, and all the different vesicles. So you can look at the green channel, like you can see there's a lot of like tiny little dots. We can actually, what's really cool with the 3D cell analysis is that we can actually try to use that to generate both the detection of the whole cell and also for just for the individual vesicles. So first thing I'm going to do is I'm going to define an ROI so we can do the preview and take a look, do a preview and process the data a little bit quicker. So turn on the eye, and then also make sure you click on this little box here to apply whatever selection you're working on, to apply whatever settings you're working on the recipe to just that ROI region. And then of course, when you're done with the analysis, when you're done with the ROI, make sure you turn this off before you click start so that you can actually apply this to the whole image. And for this one, we're going to try to adjust the box a little so we can incorporate just one of these cells. So you notice that I'm actually only picking the dimmest cell out of all of them. And the reason for that is because I actually want, for the automatic parameter determination, I do want to make sure that all of my cells get detected. So if I pick the one that have the brighter object, there's a good chance that I might not actually get the parameter that I want and would have to change it. And then of course, kind of forgot about this. In the input and output, very important, make sure you have the right settings on this. So I'm going to change the cell to the green channel. The nuclei is still the cyan channel, which is fine. And then going to do the green channel again, then the red channel, which you can see here. And then finally, I'm going to add another vesicle detection. If I click on this gear icon, I can actually click on the plus, and I can add an extra group for the vesicle detection. Can get rid of it if I don't have anything else to detect. I can also turn off the nuclei if I want to. But for now, I'm going to add a new vesicle group so that I can detect also the magenta channel as well. And when you're doing when you're using the automatic parameter determination, make sure that your ROI is centered on a single on either one object or a cluster of objects that are very close to each other. The reason for that is because the parameter picker will try to actually determine based on it's actually closer to the center of the box. So for example, if I extend this all the way out to here, the center is just going to be this empty region. And so like it will actually cause the parameter determination to kind of overestimate what the parameter should be. How are we doing so far? And again, if you have any questions, just feel free to send it in the chat or unmute your mic, or you want to show us, you can also we can also share your screen with us to show us what if you have some problems or you have something that you want to show off. So I'm just opened up the whole cell detection group, and so I might click on the preview so you can see what the parameter is going to look like.
Sorry, Julian, I'm going to just quickly interrupt you. Just because Alex is asking if you can quickly show the input and output sections of the recipe again.
Oh, yeah, sure. Yeah, so for the input, the cell is going to be 488A, and then the nucleus 405. Then for the vesicles, we're going to do the 488, 561, and 640.
And to add more vesicles, Alex, that cogwheel right there. So that number at the bottom next to the vesicles, you can add more or decrease it and whatever you want.
Yep. All right. So this is the preview of the with the automatic parameter determination. The cell. So right now the preview has a transparency on the surface, so it can be kind of hard to see. If you want to change it, go to the alpha column in the object set settings, and then change this transparency slider down all the way to the right. And I'm also going to turn off the channel so that I can see this more easily. Okay, so in this case, I think the parameter looks pretty good, so I'm going to leave this as is. Now I'm going to open up the nuclei detection. Sorry, it might be easier if I close the input and output. So Alex, are you okay if I close the input and output section?
Yes, sorry, I caught up.
Okay, great. All right. So next thing we're going to do is show off the nuclei. I think we can, same thing. So in this case, I think we might be a little bit over segmented. So I'm going to actually change the nuclei intensity to a little bit higher. So I'm going to go with 4,200, but you can play around with any number that you like and see what you like the best. And so you can still see kind of it's a little bit bleeding out to the edge. So I can also just increase this further. And one thing that I forgot to mention in the presentation is that you can also adjust this. Let's say if your detection, if you're trying to detect an object that only has a membrane stain, for example, you can use the fill hole size parameter to actually fill in the gap between the membrane so that you can actually get the whole, so you can get detection of the whole object. So it's a pretty useful tool, especially when you're imaging things that are not always, that doesn't always have like the full intensity. I think this parameter is also pretty good. So the next one I'm going to do is I'm going to go to the vesicles 1 detection. And so you'll notice very likely, so this is the back to the green channel again. So you notice that this is going to be way over segmented for what I'm trying to do here. So actually, I'm going to extend this out so I can actually see the second object. So in this case, like since I'm not using the automatic parameter determination, I can change the size of the ROI box there. And let's change the transparency back again. So again, like you'll notice that it's really over segmented in this case. And so I'm going to actually, I'm going to change the object background removal to change to remove background so that I can actually clean up the cells a little bit. And you can go back to, if you go to the image, so go to 2D, you can use the annotation tool to measure how big each of these vesicles are roughly. So it's like 0.72 or let's see, 0.8. So I'm going to keep this at 0.5 right now, just to make sure that I can, just to cover all my bases. But let's see if this is, no. So here, like since I've changed the background removal parameter option, so I can play around with the intensity until I get the result, or I can also just do the automatic determination again. But just keep in mind that when you do the when you click on this automatic parameter determination, it will apply to every detection group on in the recipe. So if I click this right now, this will actually change my nuclei detection back to what it was determined previously. So just be aware of that. I think I actually might need to still have to do this because I actually don't know. Let's see. Okay, so now we've gotten most of the objects segmented. It's a little bit it's a little bit aggressive, but it's just again like just with this just with this automatic determination, you can get to a result that is kind of close enough that it just needs a little bit additional fine tuning, and you'll get to the result that you're looking for. Okay, how are we doing so far? Good. Okay. Again, the object is kind of over segmented, so I'm going to increase the vesicle intensity. So a little bit. So with this, the 3D cell analysis, a lot of it is like there's like some amount of iteration that that is going on that you need to kind of play around with to try to get the best detection parameter. Kind of similar to like the other recipe, but only this one has like a lot more options. So if you have any questions, feel free to ask. I'll try to explain what the parameter means and yeah, and how to set them better. So. And also for this one, I don't really since it's pretty since the objects are fairly continuous, I'm actually going to do the skip partitioning so I can actually get the connected objects all looking like it's a single piece. Yeah. And then finally, we'll work on the 640 channel. Get the result. And same thing, I might need to do the to change it to the remove background. Let's try 200. So yeah. So just one note is that the background removal is a pretty aggressive algorithm. So when so as you see on the original data, it was actually the intensity determination was like about 5,000 or 6,000. But when I actually added the remove background, my min vesicle intensity even at 200 was barely giving me any detection results. So just a good thing to keep in mind is that like whenever you apply the background removal, you will need to drastically lower your the vesicle or the intensity parameter so that it can actually generate the detection. Okay, so now that everything is, I think all the parameters are pretty good. I'm going to hit start, and this will apply. I'm going to apply this whole analysis recipe to just this ROI here. So you'll see that I have this cell would be my in the result, and then so there are two different output options that you can choose in the 3D cell analysis recipe. You can either select the meshes recipe, which generates morphologically accurate mesh output on the vesicles, or if you just wanted to do get the count and the distance of these the approximate distance of these objects to the centroid, you can always change them to. You can always use the spots recipe, which will not give you the morphological shape of the objects, but it will just and every vesicle will just appear as like an individual little sphere. So here's the result of the analysis. So I'm going to turn off. So the output is actually color coded a little bit so that based on the color of the channel. So the cell membrane is green, and then the nuclei is in this turquoise. And then the red and the magenta shows you like which the vesicles the channel belongs to. So, and then here we also have the mesh transparency, so you can actually see through the cell membrane. Or if I want, you can always just make this whole thing very opaque, and then can't see anything. So one thing that one thing that would be good when it comes to the analysis too is that like you notice that the object looks very blocky in the result, right? In the results right here. So like if you have ever played, I don't know, like Minecraft or like SimCity 2000, you'll notice that like everything looks like there's like a kind of like a flat level, and then like it just kind of like slopes down and then slopes down, so it looks very blocky. So to change that, you can change the mesh smoothing factor to get rid of that. Of course, the processing will take just a little bit longer because it will need to apply the smoothing, mesh smoothing to the detection. So yeah, let's see. And now I'm going to turn off my ROI, so I will apply the analysis to the whole image, which hopefully shouldn't take too long. So does anyone have any questions or like anything that is unclear that you want me to kind of go through again?
Okay.
Cool. Yeah, so after this, analysis, the recipe applies, I can show you guys the charts, so you can look at the different measurements that are available in with the recipe. So were you guys able to get results on your with your analysis, or do you guys want to show me or what your analysis looks like or anything? Just let me know.
So I managed to get it right, except I didn't realize that the cell that I picked didn't have any vesicule 3 in my ROI. I just picked one of the cells that didn't have any of the magenta vesicles, so I'm redoing it for the whole volume. But otherwise, I'm good. I'm back.
Okay, great. Yeah, definitely like the ROI, like setting the right ROI for the parameter determination is it's pretty important in this for this recipe. Or the alternative is that you can just turn off the ROI and do it on the entire image. Like since this is a relatively small image, it wouldn't take too long. But like imagine you have a data set that is a couple of gigabytes or even like tens of gigabytes. You probably don't want to use that automatic determination on the whole field because it will take a long time because it has to process the entire image and to try to find the best parameter. So and of course, like so for in the workshop, like we can use the small field, but like on your actual analysis, you might not want to be waiting for like 15 minutes just so you can get a parameter. So the ROI, it's pretty useful in that sense. Okay, so if we go to the charts, we have a lot of different measurements that we can plot with the relationship. And so you can see like all of, we have a whole list of measurements. So we have things for sphericity, for all of these like different object sets. So I'm going to actually plot, let's look at vesicle 1. So it would be this one right here. So this, we changed the color so that can make it more obvious. Yeah, okay. Yeah, so this yellow object set. So I'm going to plot maybe the sphericity to, I guess, the distance to the nearest edge of the cell, for example. So, nope, did I have the right one? Nope. And so you have like a lot of different options to look at that. You can also change the color of the object set, so to make it color by measurement. So for example, so here I'm going to change to distance to the nearest edge of the cell. And so it's now color coded based on how close it is to the cell membrane boundary. And I can change the slider so you can see right here that all of these objects that are kind of like in the red, it's like further away from the edge of the cell, and then like everything that is blue, it's it's pretty close. I'm gonna actually turn off the cell membrane so you can take a look at that. And then another cool thing that I could also do is to change the parameter to maybe the edge of the nucleus. So this is another different way that we can, a lot of different ways that we can show visualize all of the objects. And of course, like all of these are measured together. So you can, sorry, I have keep moving this window. Can I hide this? Okay. So you can also get a count of, say, for example, how many, so let's say for this cell, how many vesicles are in this in this object, or same thing, I can go to the nuclei, and then like there's also like a different count on number of vesicle count within the nuclei itself. So do you have any questions? Or can I move on to the next part of the demo? All right, I'm going to assume that we're all good. So let's move on to the next demo. And if you have any questions, again, just feel free to unmute your mic and ask the question, or you can also send it to us in the chat window as well. Okay, for the next demo, I'm going to go back to File, Open again. And this time we'll select the Drosophila egg chamber image. And in case Avia like kind of like lost its location, just make sure that you go to the you can find the demo data in the shared data folder under demo data and 3D MSA. Okay, so I'm going to just a little bit. So for this one, I think you, so this is one of the images we have shown on the video, on the presentation previously. So we have this whole object, and then we have a stain that actually just labels the membrane in green. And then we also have the individual nuclei that are in blue on this one. So for this one, what we're going to try to do is to actually detect the whole egg chamber as one object using the green channel, and then we'll pick up each of the individual nuclei as a vesicle in this case. And so now we'll go back to the recipe console. Let's go to the input and output. Since we only have 2 channels and we're only going to do the whole cell, the whole egg chamber and then the individual cells, I am actually going to click, so let's click on the clock. We can turn off the nuclei by clicking on the checkbox, and then we'll click on the minus so they can, let's keep to 2 vesicles. So we'll try to detect the individual nuclei as a vesicle in this case, and then also the membrane in the green channel as the other vesicle. So let's change to this. And of course, this image is also relatively small. So if you want, you can also try to create an ROI to do the parameter determination. Or you can also just use the whole image. It might take a little bit longer. So let's do that. Okay. You guys following everything so far? Everything's good? Okay, great. All right, I'm going to turn off the blue channel. So for this one, what I'm trying to detect is actually the whole egg chamber. So this would be from the green on the green channel. This is basically what the whole thing looks like. Basically, it looks like an egg, right? So in just that's the slice of detection I have, notice a couple of things. So, excuse me. So first thing is that the inside is not detected at all. And the other thing is that it is already cut into 3 pieces, even though I want to detect the whole organ, so the whole egg chamber as my whole cell, in this, as the whole object in this case. So 2 things that I'm going to do. I'm going to change the min cell intensity to a lot lower, so that I can try to get the whole, get the segmentation of this middle part. You can also change the whole size to fill it in, but it might just be easier if I just do the cell intensity instead. And then the other thing I'm going to do is I'm going to turn off the partitioning. Since this whole since we're only looking at one whole object in this case, there's really no need to apply partitioning. It just kind of slows down the process a little bit. So this is what it looks like. And so I'm going to actually, so in this case, I'm actually going to try to do the fill holes for this one. Let's see. So if we go back to the 2D image, we can actually use this to measure how big, what the area is. So the fill holes is based on a 2D cross section of the image. So if you want to know what the fill hole area, just use in the annotations, use the freehand region tool and just kind of draw kind of a box around it, kind of approximate. So this area is about 2100. So I'm going to be at 2000. And then I'm going to go back to my 3D image and see if this actually works. Probably a little bit too well because I have a small ROI. Oops. Okay. So let's just. So maybe I'll just turn off the ROI so we can actually see what the parameter looks like. Since this is a pretty big object, it will take some time to actually to run. Okay. All right. So now so this is the whole, like what it looks like on the whole cell. So pretty good, I think. Like you notice that it is a little bit bumpy. So like the other thing, like what we can do is we can just refine the cell intensity and the smoothing factor a little bit so that towards the end, like when we're finished with applying this on this image, we will get a smooth object out of it. And then I'm going to, for vesicle 1, oh, I forgot I should have done the ROI. Anyways. So remember that the parameter determination and also everything that we've done is going to be based on the processing step that is already applied to this image. So if I look at this, you can think the cells actually look pretty nice, I believe. Let's see. Let's turn on. So it is doing a little bit of extra partitioning more partitioning than I wanted to. So I'm going to go back to the 2D view and try to measure out how big each of these nuclei are. And so another thing that is different between this recipe and the object analysis is that you'll notice for the partitioning steps and also for the object filtering, it is based on the diameter and not the radius of the object. So just kind of be aware, like when you're entering the vesicles diameter, when you're measuring the object for filtering, make sure you use the diameter and not the radius. So it's like looks like about 3. So I am going to just to be safe, just do 2, and then this partitioning. This is going to be measuring the distance between two objects that are touching from the center of the object to the closest. So it's kind of similar to the radius in this one. So I'm going to do 1.5. So this is looking a lot better than before. I'm also going to just change the intensity up a little bit. Oop. That's too much. Okay, and then on the last step, we're going to try to segment this whole thing. I'm going to increase the vesicle diameter a little bit so I can get the detection, see if I can get the actual detection, because it doesn't look like I actually have any object that are being detected right now. Okay, so now you can see the kind of like everything is kind of connected. You can get the nuclei that we have actually segmented the membrane as like a whole object. So I can increase this a little bit so that I am not getting a lot of these holes filled in as well. Okay, this looks pretty good. So if you guys, whenever you're ready, so actually before I do this, I'm actually going to turn off this fill hole size just because it is currently filling up my entire object. So later on, like once we are finished, so let's see, this is kind of getting close, but maybe like. Okay, let's decrease the cell intensity a little bit. Oh. Yeah. Yeah, so in this case, you'll notice that the parameters are pretty sensitive. So actually, I think 375 is probably the closest. Looks like I got everything, and then and nothing gets like overly segmented. So I'm gonna click start. So if you like this, you can apply this to the whole this recipe, like all the settings to the whole image. It will take a little bit of time for it to do. Like last time when I tried it was about like 10-15 minutes. So, but we do have a lot, but we still have like about 1 hour left on the workshop time slot. So you want, feel free to play around with this. Oh, wait. Okay, I'm going to turn off the cell membrane. So you can see all of the nuclei and then also the boundary of this. And then I can, once again, just change the color settings. So for example, for these, for the nuclei, I can change the color by measurement to say distance to the edge of the cell. And so you see like the objects that are in red are much closer to the edge compared to the ones that are in blue. All right. So that's all of the demos I have for you today. Do you guys have any questions about anything? Is there any step I can go over again, or if you want to play around with it some more, just let me know. Just let us know.
Okay, thank you. I think I'm good.
Okay.
All right.
Great. Yeah. So I also just wanted to show you guys, like, since we have a bit of time left, so this is one of the demos. You can actually see this. So Luciana, so on the launch webinar, this is the mouse embryo image, and yeah, this is taken in an optical coherence tomography. And what the segmentation does is that we actually detected the whole mouse as the kind of like as the big object, and then individually, then we can look at the different parts of the mouse, so like the eyes, the brain, the guts, and then all of that. It's possible using this recipe to do.